Thank you, Albert, and also thank you, Diana, for inviting me to this very exciting conference. And before I start, I have to mention that, I'm coming from a parallel field, and we used to work, we're still working in the field of drug delivery systems. And nowadays, it's called nanomedicine, and I think that's a blessing for those of us who are not resident in the United States, because when we travel through the immigration in the United States, we are often asked what is the reason for your visit and what you actually do. So I don't have to actually mention anymore that I am here for a drug conference, I do drug delivery, and by the way, my last stop was Colombia. So I can tell them I'm actually doing nanomedicine, and they can understand it. But coming back to what we have been doing, I personally started in this field, goes back to mid-80s, 84, 85, that we were trying to use a broad range of particles for the purpose of drug delivery and site-specific targeting to different organs. And surprisingly, those days, although we use various kind of nanoparticles, we never call them nanoparticles. So if you go back to the old literature, you will find maybe thousands of these kind of papers, but actually referred to as microparticles, and that was the terminology those days. But what was interesting, we knew from early point that when you are trying to make a delivery system and you administer that to the body, to an animal, you are against the immune system, because that is the purpose of the immune system, to protect you from the invaders, and also not only the viruses and bacteria, but also any kind of particulate matter that is body getting exposed to. So in order to actually design this kind of delivery system, we had to learn basically from the disease processes. Are we going to make just a particulate system and try to target it somewhere, or are we going to understand the physical or the physiological basis or the pathophysiological basis of a disease? What are the opportunities offered by a particular disease, and can it really be exploited in terms of the design and site-specific targeting using this kind of wonderful materials that is available today? And basically, those days I was in Nottingham, and if you know that in Nottingham you have the Robin Hood, and that is the symbol of the targeting, and that is why I actually went to Nottingham to actually do the targeting. And those days that we started with the nanoparticles, and we knew that when you administered any kind of particles, they're rapidly cleared by the Kupffer cells or the liver macrophages, and we look into the molecular basis and the molecular mechanisms, how these kind of cells can recognize different type of particles of different properties. What are the key important physicochemical aspects that could actually be used in order to either target these particles more efficiently to the macrophages for treatment of disease and disorders of the macrophages, and at the same time, we were also trying to look at strategies how to prevent the clearance of particles by the macrophages. And by late 80s and early 90s, we were among one of the pioneering groups that actually made the stealth system and with polymeric particles, and that eventually led to the development of stealth liposomes, which coming from the stealth fighter terminology. At the same time, we were trying to exploit how you can actually exploit the pathophysiology and even make particles specific for different subset of macrophages. And because macrophages are not heterogeneous, they are very, very different, even within the same organ, so we need to understand the cell biology of the macrophages, and also the way that blood is actually flowing through different organs. And by doing that, we even try to make the very first spleen-specific kind of nanoparticle, which was reported in 1991. And then eventually we came to kind of a bone marrow-specific nanoparticle, which was selectively targeting the vascular endothelial cells in the bone marrow, and we were looking in terms of the processes, what actually happens inside. And these days we are now focusing mostly on brain endothelium, and we have designed some specific nanoparticles based on some peptides, which they self-assemble and exclusively target this kind of brain endothelial cells. This has not been published. We are trying to get few patents for the protection at the moment. And we're also going into different type of cells, including macrophages, for organelle-specific targeting, and in this case, mitochondria, that you actually see in these kind of pictures. But what is important from the medical perspective is you have to be aware that in medicine, whatever you take, by definition, is toxic. Even water is toxic. But what is important is the dose. Is the frequency of administration and also the route of administration. And with regard to any kind of these diseases that we are trying to target, such as cancer or vascular endothelial kind of pathologies, you do need a proper characterization of your drug carriers, and in this case we're using liposomes, polymeric nanoparticles, polymer micelles, etc. So we need, I mean, the good thing about today's technology, or the nanotechnology, is it offers these wonderful tools to do a better characterization of the material properties. And that is very important to look into some of these old phenomena, how these nanoparticles could interact with different elements of not only the immune system, but in general in the physiology, and how to exploit them for better or optimization in the targeting. So that is basically what we do these days, not only in terms of the kind of a more precision characterization, but also trying to look in terms of structure-activity relationship. So we're basically breaking into different components, we're making libraries of these materials, we try to build them step by step, and then look at not only the cellular processes, both at target and off target, but also in terms of the humoral immunity or the first line of the body's defense, which is the complement system. And through this kind of structure-activity relationship, we select the best possible candidates for the purpose of in vivo studies and therapeutic efficacy. And what is important at the end of the day, we are not trying to actually eliminate toxicity 100%. It's impossible. What we're trying to do is to increase the benefit to the risk ratio from a clinical perspective, i.e. can we actually enhance the therapeutic efficacy while minimizing this toxicity. And sometimes you do have to use materials which have wonderful properties, but they are toxic. But if you put into the right context, you could overcome those kind of adverse effects for the patient's perspective. So in terms of the general toxicity, I mean, we do a lot of studies before I come into the immune system that what actually happens intracellularly, because at the end of the day, you could induce necrosis, you could induce apoptosis, and most importantly, necroptosis. And we're trying to look at in terms of material properties, such as those that can induce gene therapy or siRNA release, what actually happens from the signals coming from the different compartments that the material is actually moving, such as endolysosomes to the ER, to the nuclear membrane, and all the kind of important signaling processes and stress processes that are actually involved in the cell. And we not only focus on different organelles, particularly on the mitochondria and the different electron transport properties in the mitochondria, and look how these kind of materials interfere with these specific processes. And we are looking again in a broad sense in terms of the necroptosis in this case, or halo kind of formation around the nucleus, and how these processes are linked to the material properties, and how we can go back to the design table and alter molecular structures through not only high throughput synthesis, but also from mostly kind of a nano kind of approaches these days to optimize this kind of design and assembly of those materials. Generally, I mean, in terms of, I mean, that's also relevant to what most of you guys are doing in terms of the nanotoxicology, in terms of the administration. And here I'm basically focusing in terms of intravenous administration or systemic exposure. That, I mean, in pharmacokinetics and pharmacology, I mean, you do have these ADME processes, absorption, distribution, metabolism, and excretion. And we knew, again, from very, very early point that a number of physical-chemical properties can actually influence the pharmacokinetics, such as size and even the shape of the particle, which is now becoming very important and more controllable by these advances in nanotechnology in terms of the design, and most importantly, the surface properties. And we came to this conclusion many years ago that not two single particles are always the same. The surface properties are always different. And as a result, you will see different distribution of the particles, and also with respect to different population of macrophages or any other kind of cell that you are targeting. And one very interesting aspect that I see is quite a lot of attention these days is the concept of protein absorption, and a lot of people think that is very, very important. And when you go back to the old literature, that was one of the prime subjects of the field of drug delivery systems, which has not been ignored. And when you look at those kind of data and you analyze them in detail, we came to the conclusion that in the majority of the cases, protein absorption does not play any significant role. Only you can apply this context if you put it into the context of therapeutic and the real distribution and the recognition mechanisms in vivo. For example, the complement system, which I'll come and design, basically say how it actually plays an important role. But to give you a very good example in here is that there are many small particles, such as maybe 30 or 40 nanometer particles. If you do proteomics, if you do mass spectrometry, you will not find any kind of opsonic molecule absorbed onto the surface. You come to the erroneous conclusion that, look, these particles do not induce any kind of humoral immune effect. Yet when you look at in your fluid phase, you will see these pathways that actually operate because of the surface structure, because of the geometry of the nanoparticles, they cannot accommodate on the surface. And that was established many, many years ago in the field of drug delivery. And that is very important when you have very small particles and try to actually see how these proteins sit. And I can tell you, I mean, you do kind of analysis, you will find 70, 80 different type of protein bind to a surface of a particle. But I can tell you, the majority of them do not play any significant role. You could have particles. We have shown, again, there's a group in Aarhus has shown that in Denmark that if you have polymeric nanoparticles, IgM actually sits much more in terms of the concentration, much higher quantities on, let's say, 500 nanometer particle compared to a 50 nanometer particle. But yet, on the larger particle, IgM plays absolutely no role in activation of the complement system, yet it does on the smaller particle. But that's relevance. You see, you have to put into the context of the actual particle distribution. Otherwise, saying that these are the proteins bind doesn't mean anything. And to give you another very good example, that when we designed the stealth system, we were quite intrigued by the movie The Hunt for the Red October, because remember, that's a stealth submarine, right? And we were also interested, can a stealth system actually be recognized eventually by the body's defenses? And the answer is yes, because that depends on the state of the responsiveness of the macrophages within a particular pathology or within a particular stage of the development. Even newly recruited macrophages to a specific organ have receptors which are under control, and the expression is under the control of the microenvironment, which actually missing on the normal quiescent macrophages that actually recognize the stealth system, independent of any opsonization processes and protein binding. So these are the important aspects from the medical point and drug delivery, how to design system, which I think also applies to nanoparticles in a broader context of nanotoxicology. And that is a very interesting example that here we have these stealth particles exposed to plasma of two animals, one under normal condition, one under a pathological condition. You can see more protein bands actually sitting on these particles, yet both these particles behave as long circulatory. So where is the relationship between this abnormal protein absorption and the distribution and the pharmacokinetics? Absolutely none. But then you have to put that into the context of what particles you are using and what is the application of that particle or the end point of that particle with regard to, let's say, a therapeutic field or any other thing that you like. But what is very intriguing, that there are a few number of nanoparticulate systems or therapeutic particulate systems that reach into the market, approved by the regulatory bodies. These are some examples. And surprisingly, a selection of patients that receiving these kind of materials show symptoms of hypersensitivity or kind of an anaphylactic reactions. And basically, during the infusion, within a few minutes of some of these materials, you will see that the patients starting to react. And what happens, they get chest tightening, their lips start to swell, they get the feeling of suffocation, blood pressure starts to drop, pulmonary pressure rises, and if you don't give them the proper medication, they probably have a couple of minutes to leave. So as a result, with all this kind of formulation in the clinics, they give them steroids pre-medication, they give them antihistamines, and even the rate of infusion is very, very important to control this kind of reactions. And yet, you do have materials like this kind of Taxol, which is paclitaxel solubilized in this micellar structure made from cremophore-EL, which is an ethoxylated castor oil, that in spite of giving these pre-medications to the patient, there is a fatality of about 2 to 3%. So 2 or 3 individuals out of 100 actually die during the infusion of these kind of materials. But not fortunately with the liposome has not been reported. And this is a field which actually pioneered by one of the audience, Janusz Zebeni, sitting there, and he's basically responsible for dragging me into this field, which actually relates to the mechanism why this kind of nanomaterial is actually doing this. And then we started to work in this area. So the culprit of this, we think, is the first line of the body's defense system, which is the complement system. And if you are not familiar with the complement system, this is the basically immunologically is there to protect you, and you could trigger this by 3 different known pathways: the classical, the lectin pathway, and the alternative pathway, and there are different initiation molecules that could bind to the surface of the particles and trigger the whole cascade. But what is key in here is this molecule which is called C3, which is not only responsible for the opsonization of the particle and the clearance by the macrophages, but its cleavage through all these kind of enzymes and kind of molecules involved in the triggering of the complement is you are releasing a small molecule called C3a and eventually another one called C5a, and these two are anaphylatoxins. So they are really rapidly cleared by the body, but sometimes they could actually induce this kind of anaphylactic reactions that we see in different individuals. We don't understand the actual mechanisms. There are some immunogenomic factors involved that we are looking into it, but the point in here is why different materials of different surface properties or surface chemistries can actually trigger this complement. And okay, that is quite obvious because you do have antibodies or kind of molecules such as the C1q that actually could sit on the particle surface and do this kind of triggering. And you also have the fluid phase, which is basically is a water molecule actually causing the activation of the alternative pathway continuously. But what I would like to draw your attention today is to the role of this lectin pathway. Now, lectin pathway evolutionarily evolved for the process of microbial recognition and recognizing usually the sugar groups on microorganisms and particularly the mannose, right? And yet, I would like to emphasize in here that the majority of the nanoparticles that we are actually using do not express mannose groups. And what I want to show is the importance of the lectin pathway in terms of these anaphylactic reactions and activation of the complement. So this is the C3 molecule. When it's activated, you're forming this secondary structure called the C3b, and that's the process of the opsonization going on the particle surface with the gradual release of the component and eventual clearance by the macrophages. And again, what is important, when you look at the crystal structure of the C3b and when you look at how it actually accommodates on the surface of a nanoparticle, one single C3b molecule occupies a space of 40 nanometers squared on a nanoparticle surface. So if you take, let's say, a liposome of 100, 150 nanometer, you will find plenty of C3b covalently attached to it. But if you take a gold particle of 20 nanometer, you will hardly find the C3b on the surface. Yet, you will see potent activation of the complement system because it's all in the fluid phase, and you have missed it because you have not measured what is in the actual supernatant. And that actually happens because of the alternative pathway, and the C3 convertase of the alternative pathway is in the fluid phase, not on the particle surface. So these are the important aspects that one needs to look in terms of recognizing or characterizing the particles and their immune responses, or even in terms of the protein absorption. So basically, when you're releasing these kind of anaphylatoxins, they will act on a large number of blood cells, such as macrophages, neutrophils, monocytes, and so on, platelets. And that caused the release of all these kind of secondary factors, such as histamine and leukotrienes, and very large number of pro-inflammatory cytokines, that you eventually see this kind of adverse effects on some specific patients. And that could be maybe due to the threshold activity of the receptors, which this anaphylatoxin or this kind of pro-inflammatory cytokine actually operates in these individuals. Also, you have to be aware that the coagulation pathway itself could trigger complement system directly and by cleaving the C3 and generating the anaphylatoxin. And again, that is a very important case when you are dealing with cancer, because you do have a lot of blood clots in human cancer, and you do see complement activation in the cancer, and I will come back to that for an important message. So what we are trying to do these days in the lab is to actually understand this kind of interaction going between the size, the morphology, and the surface characteristics of the particle. And you could break them down to all these kind of parameters and trying to map what is actually going on in terms of the complement activation. And I can tell you from about 20 years of experience, if you do that, you come absolutely to no conclusion. This is a net effect. This is a combination effect of all these parameters. And in order to balance that for the purpose of the field of drug delivery is a very, very challenging area. I mean, never mind the nanotoxicology or how a specific particle can actually trigger that. So you have to look at that from a much broader perspective and through an integrated approach. What is the relation between the size and the morphology? Because if you take a liposomes and you load the liposome with a specific drug, you can actually change the curvature, and maybe from a spherical shape it becomes to more oblate shape. And those stress areas on the membrane could be the actual triggering site of the complement activation. The same applies to many other nanoparticles or even the coating. So what I would like to illustrate now, some of these important properties in relation to nanoparticle engineering and some of the responses that you see by giving you 2 or 3 very interesting examples that we published just last year or coming this year. Here we have used a polymer which is called poloxamine 908. You can see the center is polypropylene oxide because it's hydrophobic, could sit on the surface of a hydrophobic particle like a polystyrene. And the good thing about polystyrene is you could get them in a very near monodispersed kind of preparations. And here we have used a particle of around 230 nanometer with a very low polydispersity. So we characterize this. So we're basically putting this polymer on the surface. We're measuring an absorption isotherm, and we are looking into the thickness of the polymer layer by laser light scattering. Then we're looking in terms of the thickness, and you can see that you come to a 2 plateau region, around 4 nanometer thickness in here and 8 nanometer thickness in there. But when you analyze in terms of the number of the molecules which are sitting on the particle surface, you will see about 3 times more on the top plateau rather than at the first plateau. And when you do kind of an advanced rheology or viscometry, you can actually see the viscosity starts to go up as you're coming towards the first shoulder, and then it starts to drop. And that was an interpretation of the polymer conformational change, especially the ethylene oxide. So you're having a kind of a brush conformation right at the top, while at the early stage you have a kind of a flat conformation. Because of the crowding and water repulsion, then the change starts to expand from the surface. We have actually proven the same thing by atomic force microscopy. I don't have the data to show you in here, but again, as a kind of a nanorheology, and again, you can see in terms of the scanning electron microscopy, you can see that you have the aggregation of the particle as you come to the first plateau. You are making hexagonal phases, and as you go on top, you're actually trying to stack that because of the electrostatic repulsion. We also characterize the polymers itself in terms of the polydispersity and the purity and so on. So basically, we have a big combination of different expertise in our group that they actually do all this kind of analysis, and we put the whole thing. But what I would like to tell you that these particles, if you make them into this polymer conformation around 230 nanometer, they are not only long circulating when you administer them to an animal, but they exclusively target the spleen. Okay? I'll come back and explain why it actually happens. But what is very important in terms of the complement activation is when you look at the polymer conformation, regardless of whether you have polymer on the surface or not, and we're talking about the long circulatory particle, you always see complement activation. But remarkably, changing the polymer conformation from a flat configuration to a mushroom brush configuration actually changes the mechanism of complement activation. And that was the biggest surprise that we saw, and that was published last year in ACS Nano, and also gets a news and views in Nature Nano regarding the importance of the polymer conformation and nanoparticle design for the safety aspects. Now, when you don't have a brush conformation, the activation you can see is through the C1q, which is the classical pathway, and also through the alternative pathway. But when you come into a brush conformation, you can see the role of the lectin pathway, which is becoming very, very important. Yet, we do not have any mannose structures in here. Right? So the question is, why a simple change in polymer conformation can alter the immune response or trigger the same pathway, but yet through a different mechanism? The answer is very simple. This is mannose. Right? Look at the structure of the end chain of the polymer. Transiently, it actually represents a section of the mannose. But if you have chains of these polymer, which are basically moving continuously, you are transiently structuring a kind of a mannose on the surface. And there are 2 regions in the mannose. Basically, one is these 2 axial hydroxyl groups, which is a template for binding of the mannose binding lectin protein, which actually causes the triggering of the complement activation. Also, the top region we have found is a binding region for another molecule called phycolin, L-phycolin, which also sits in there and triggers complement activation. So basically, you could see, you could use a material which is considered neutral from a chemical point, yet on the surface transiently resembles mannose, and you are converting a small particle into a pathological particle like a bacteria. And that is how the immune system actually sees. So basically, the immune system sees in here the pattern recognition. So the pattern recognition becomes very, very important, and is actually repeated on the structure of many nanoparticles. Again, to show you some of the pharmacokinetic data, you can see that the particles after a few hours, you can see the liver actually goes down as you come into the top plateau, and the particle goes to the spleen. And that is the reason is because in the spleen, the blood flow it goes to the red pulp. There is no direct connection between the arterial and the venous circulation, so the blood is exposed to the entire tissue, and that is where you do have the macrophages, right? So basically, we were interested what actually happens to these kind of particles which are resistant to the clearance by the Kupffer cells in the liver, what actually happens when they go to the spleen. And because in the spleen, these are the venous circulation, you have the endothelial cells, and the gaps between them are in the region of 200 nanometers. So if your particles are rigid, like the one that we have made, and if they are long circulatory, i.e. not recognized by the liver, they will be filtered at those interendothelial cell sleeves and remains there. But the answer, the question was, can they be recognized by the macrophages? And the answer is yes, because that is the cross-section of the venous circulation. These are the endothelial cells, and you can see that the macrophages there, and they're packed by these kind of nanoparticles. Yet you do have the steric barriers. And what was surprising again, we have found that the process of complement activation had nothing to do with the recognition of this particle by these macrophages. Again, shows the concept of the macrophage heterogeneity and the control of the macrophage receptor expression under different microenvironmental conditions. So you cannot come and say that I have a long circulatory particle, because it's not within the context of immunology. So you need to define what are you trying to do and what cells you are dealing with at the right microenvironmental conditions. In a sense, we have copied the malarial parasite in here in terms of the design. These are the red cells, and these are the parasitic inclusion in the red cell. And what happens, these red cells is deformable, will go through these gaps between the endothelial cells. This part is rigid, around 200 nanometers, pinches off and remains in the spleen. And the macrophages will take that kind of inclusion, and that is why you get the splenic infection from the malaria and the enlargement. So in a sense, they have copied malaria parasite for design of a spleen-specific nanoparticle. Another interesting concept, because pegylation has been, in the field for many years. We have started the field of the pegylation, apparently one of the pioneering labs. And now the PEG phospholipids are used to stabilize carbon nanotubes, right? So medically, I cannot say that the carbon nanotubes are really advantageous over anything else. No, I'm not working in collaboration with Janos and the US Army at Walter Reed Army Institute. And we found at that time that this activation by PEG liposome is different. Now, we are talking about the same material, yet the molecular basis of activation is different, because this PEG phospholipid, they have an anionic charge. And we have found that this anionic charge was responsible for complement activation by doxyl, but replace it by a methyl group, and you can see the activation totally disappears. We can change the quantity of these PEG phospholipid into these liposomes, and you can see there is no activation when you do the methylation. And the molecular basis is very complex, because activation is exclusively through the classical pathway and alternative pathway. So basically what we are doing in here, we are sterically generating a methyl group on the binding side, because these kind of PEG phospholipid molecules are around 1 nanometers above the plane of the phospholipid bilayer. And there are some antibodies which have 2 binding sites. And if the orientation is correct and the topology is right, they actually sit in there, and then they accommodate C1q and activate complement. If you make the steric barrier, the antibodies actually sit on the membrane, but they cannot activate complement because they're not in the correct posture. At the same time, the PEG could actually make some hydrophobic interaction with the C1q, and that also causes the triggering of the complement to some extent. So you can see that the molecular basis of activation is different when you change the particle shape or the particle composition, yet using the same material. So it's very important from the immune system, you actually characterize this in a more thorough way and try to see what actually happens in terms of targeting and biological distribution. So this is the last slide, and coming back to the cancer, remember doxyl is for cancer treatment. Now, these kind of particles can trigger complement activation regardless, and they actually go to the cancer and they accumulate at these interstitial sites, not taken up by any cells. These are fluorescent labeled, and usually at the capillary roots of the angiogenetic vessels. What is interesting is there's a paper came about a couple of years ago by Jan Lambris at Philadelphia, showing that complement activation in the tumor can cause tumor growth, right? So the question we ask, by using this kind of treatments, are we really treating the tumor, or we are not helping the patients, and we are trying to make the tumor to actually advance and grow even better? And when you look at it from a clinical perspective, you do have a lot of blood clots, as I mentioned earlier, in the human tumors. And thrombin can actually cleave complement protein, the C3, generating the C5a fragment eventually. And the C5a fragment is chemoattractant to FOXP3 cells, which are the immunosuppressive cells. They home to the tumor, suppress the immunity, the tumor starts to grow. So that could be actually happening with many of the nanoparticles that you are exposed with, and that may be actually inducing tumor growth maybe on those individuals who actually have cancer. So that's a very serious area that we have started to look into it and see basically what happens from the complement activation within the tumor itself. So finally, these are some of the main collaborators that have contributed to this work, and especially I have to thank Janusz Beni, who is sitting there by dragging me into this field and make my life misery for the past 10, 15 years by introducing me to the complement. I hate it. And also the funding bodies, especially the Danish Strategic Research Council that has funded a massive center in our university to actually look at the structural activity relationship between the materials and the nanomedicine side of it, and also the FP7 program that we are part of, the NAT project, looking into the Alzheimer's disease and nanoparticles. And thank you very much for your attention.
Thank you very much for that very new take on a very old system. I think we have time for a couple of questions. Victor, please.
I wonder about whether there is experience we could see or something similar about memory against the objects and against the back or visualization.
Sorry, you need to repeat. I mean, I cannot hear you. So
About memory. I don't know if there is some repeated injections of Doxil. If there is, probably has been tried. There is memory against the carrier, against the PEG, antibodies against PEG after repeated or not?
Yeah, that's a very good question. Yes, there are a lot of studies on that. When you give repeated doses, two things can happen. First, you could induce antibody formation regarding the surface properties of the material, or the PEG in this case. And that has been demonstrated. And I can tell you that 25% of normal population, regardless of prior administration of Doxil, they do have anti-IgM antibodies in their blood. Right? So if you take 100 people, you will find 25 people have antibodies against PEG. So that is one important case. So that is why not all particles are long circulating in everybody's whole population. With Doxil, the question with Doxil is the doxorubicin, because doxorubicin is cytotoxic. It actually kills the B cells in the spleen, so you do not see antibody formation. Right? So in this case, the drug is actually helping you not to form a kind of an antibody to trigger complement or any other kind of case. But yet, the doxil structure can trigger complement activation.
Further? Here, please.
Yeah, I want to come back to the point that you said several times that the protein bound on the nanoparticles, and let's focus on complement proteins, has no effect on the issue on the cells. So that's very interesting, but the complement system has complement protein has various numerous receptors on various kind of cells, so it's a very wide range, and it's known that the interaction of some component with some receptor on some kind of cells are either activation effect or inhibitory effect. So I think it's a field which is very large, and I want to have some precision of what you actually saw and what you guess.
Yes, I mean the concept of the protein adsorption, I think it's a very sensitive one, and if you look, we have just published an article in the Annual Review of Pharmacology this year, which we heavily criticized the concept of the protein adsorption, what it actually means. And you have to be very careful in here, because if you are looking what is adsorbed onto a surface of particle as whole, you have a population of the particles, you're doing the experiment, and you're telling me that, look, there are 70 type of protein adsorbed to the surface, right? Now, the question in here is, you are measuring a bulk effect. You are not looking individually, and you may have, let's say, 5% of your particle population express a kind of a chemistry that absorb 3 or 4 of those proteins, but not on the rest. And you're coming to a conclusion that, look, this protein is everywhere, which is not correct. It has to be within the context. If those proteins are sitting on the surface, are they biologically active? Are they pharmacologically triggering any kind of mechanisms? In a way, the protein adsorption is a kind of a safety mechanism by the body to inert the surface, in a sense. And when you look at the old liposome field, I mean, we have shown that there are many important proteins that would bind to the surface of the liposomes. And some of these liposomes actually show a specificity for certain population of the macrophages, i.e. we're looking for a hierarchy system. And the protein adsorption is a kind of a dynamic process, i.e. you are removing a fraction of the particles earlier through one specific receptor, yet the surface modification allows recognition, subsequent recognition, maybe by the same macrophage or a different type of macrophage later. So it has to be within a context. If it's not within a context, it does not mean anything.
Okay, last question. Please.
I use yours. I come from infectious diseases, and complement is one of the major mechanisms for defending us from bacterial infection. There are bacteria that use up to 70% of their genome only for inhibiting complement. I was wondering whether we may actually learn from bacteria the mechanism of the escape that they use for cheating complement, how to make better nanomedicines.
Exactly. I mean, that's a very, very excellent statement, that there are a lot of work being done at this front, that learning from the pathogenic microbial kind of systems, how they overcome the process of complement activation and how to graft these kind of materials to the surfaces. And one good example is factor H. And that has actually been done, again, by John Lambert's group, that they've attached to the particle surface, and you do see a substantial decrease in complement activation. And basically, going back to early 80s, the reason why we use this kind of polymers to modify the surfaces, that was a strategy learned from the pathogenic microorganism in terms of the surface hydrophobicity and hydrophilicity. So we didn't want to make it very complex at that stage, but just looking how the simple physicochemical principle actually works. But yes, you are absolutely right. There are a large number of complement inhibitors available, such as recombinant C1 receptor protein, that we are even using some of these kind of materials to actually graft onto the surface. Yes, I think this is the future, rather than coming and do complicated surface engineering with these kind of polymers or any kind